We are Manipal BioMachines from MIT Mahe, and we're here not to find Nemo, but to save him with our project Amphiphil, a sustainable solution for vibriosis in fish. The pisciculture industry forms an integral part of the Indian economy, employing a large proportion of the Indian population and thus contributing significantly to the GDP of the country. However, excessively industrialized procedures and over-exploitative methods have led to establishment of bacterial disease epidemics, causing huge economic losses at the global and national level. Among these bacterial infections is vibriosis, caused by the Vibrio species, especially the Vibrio parahemolyticus bacteria. The prevalence of Vibrio species and Vibrio parahemolyticus can be seen in freshwater fish as per this study. Vibriosis causes necrosis of the skin and appendages, body malformation, delayed development, and eventually mortality. It was observed on increasing concentration dosage of Vibrio parahemolyticus, an equivalent increase in mortality. The current solutions include antibiotics and vaccines. Antibiotics, however, have been known to cause antibiotic resistance, leave residues that accumulate over time. Vaccines are known to be uneconomical and labor intensive in application. This is where a solution comes in. We aim to address the pathogenicity caused by Vibrio parahemolyticus using a novel antimicrobial peptide. The vibriosis infection follows in a particular way. In the presence of stress factors like high temperature, poor water quality, overpopulation in the tank, the fish becomes more prone to infection. The bacteria performs chemotaxis towards the host cell surface and adheres to it, causing hemolysis and subsequent hemorrhage and eventually mortality. What is MAM7 and why did we choose MAM7? MAM7 is multivalent adhesion molecule with 7 mammalian cell entry domains. It's an outer membrane protein which is present in several gram-negative pathogenic bacteria. It mediates one of the primary steps in infection, the host cell adhesion, and is also involved in initiating other host signaling pathways. Fibronectin, present in the extracellular matrix, and phosphodetic acid, present on the epithelial cells, interact with MAM7, forming a tripartite complex. Thus, we decided to mimic this particular interaction. Since the structures of fibronectin and MAM7 were not well established in literature, we decided to predict them. We predicted the structures of MAM7 and the heparin binding domains of fibronectin. We then performed docking studies to identify the interacting residues. The peptides were then built by mimicking one of these interacting residues. The next step was to increase the binding affinity of the peptide towards MAM7. For this, we used two approaches, an observational approach and a random approach. The observational approach involved identifying and observing amino acids manually and substituting them, thus increasing the affinity of the peptide towards the membrane protein. The second approach is a random approach that involved alanine scan, thus generating a pool of peptides. For the random approach, we used our very own software that performed an alanine scan on the input peptide, identified amino acids that could increase the affinity, and performed random substitutions, generating a combination of peptide. This gave a pool of peptide for the wet lab to choose from. This formed the design, build, test, learn cycle of our project. We designed the peptide, predicted the structures of it, tested it by subjecting it to docking, mutated to increase its affinity, and performed the cycle all over again. We also performed molecular dynamic simulation on the peptide, and we observed no conformational changes within 100 nanoseconds of the simulation. The parameters used for this simulation were based on an ideal aquaculture system.
After obtaining the sequence of a peptide in silico, it was now time to develop a strategy to produce it in vitro. Given the unstable nature of peptides within the bacteria, it was necessary to make use of a fusion system. Our vector design comprises of a peptide incorporated within an SSP GYRB split in time using Gibson assembly. To stabilize our peptide, here our intine could serve dual purposes. by performing self-cleaving ability at the end terminus and thereby reducing our peptide purification steps for the future and also stabilizing our peptide. This would further be incorporated within our vector PET22B+. Once the desired transformants would be obtained, they would be subjected to peptide purification steps as shown. Now that our peptide was ready to rescue the fishes, how would it make its way to the fishes safe and secure? Here's when we needed a delivery vehicle. Out of all the drug delivery methods available, we chose chitosan nanoparticle encapsulation to be the most viable for our project. Chitosan in general has gained great attention in the field of drug delivery due to its properties like non-toxicity, biodegradability, biocompatibility, etc. Chitosan is a derivative of a compound from the seafood industry and hence it is cost effective as well. Our implementation design, we designed it in such a way that it would be applicable to the real world as well. Our peptide would be loaded into the chitosan nanoparticles, and here the hydrophilic part of the peptide would interact with the chitosan, whereas the hydrophobic part would be bound by hydrophobic interactions. These nanoparticles further would be loaded onto a PVC mesh, which would be subjected to the inlet of the tank. With regards to the practical aspects of our project, in light of the Blue Revolution program by the Indian government, we designed our implementation setup such that we would subject a stack of meshes loaded with our nanoparticles containing the peptide at the inlet of the tank from the reservoir to the first tank. Given the flow rate in the setup, the peptide would be gradually released via diffusional effects. In order to derive the dosage of our peptide for administration, we constructed a model IC50, standing for inhibitory concentration 50. The derived equation represents the concentration of the peptide required for 50% inhibition of MAM7. As for the future aspects of our project, we plan on optimizing our peptide production strategy, performing toxicity tests to check for the plausible effects of our peptide on marine life. We want to optimize our delivery mechanism and possibly broaden our solution to other MAM7 bearing pathogens. As for our stakeholders, they belong to different categories and fields. They belong to fisheries, fish farmers, marine biologists, pisciculture researchers, NGOs, and associations that deal with aquaculture welfare, the Food Safety Committee, environmental scientists, etc. We are a group of 17 synthetic biology enthusiasts who aim at making the aquaculture system sustainable and save Nemo. Thank you.